All right, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola, and ni hao, konnichiwa, and 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 127th seminar, and I call it on MIT Day. Today is a Korean holiday remembering people who sacrificed themselves for the nation, similar to the Memorial Day in the United States. I am joining you all from, of course, the Korea at midnight today. And so actually, technically, the holiday is over because yesterday was the Korean Memorial Day. I have been enjoying my time here since the 5th, the yesterday, hearing my first invited talk out of the upcoming 15 talks during this 36-day long non-stop trip without coming home, chairing 5 sessions in the conference and also attending 3 conferences and visiting around 20 cities in 3 countries, or 3 continents actually. So I'm actually very excited. Also hope to see many of you, including the conference like SEED conference in Atlanta. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Jacquin Niles. He is a professor at Massachusetts Institute of Technology, in short, MIT. He is a bioengineering professor at the same department as that of my postdoc advisor, Chris Boy, who is the department chair now. I really thank Professor Niles especially because I do not believe we met each other in person in the past, but he accepted my talk invitation without hesitation, showing his dedication to education and nurturing young researchers and diverse community. In fact, MIT is the institute with the largest number of pioneer speakers in this seminar series that show MIT is probably the best institute nurturing and caring young generation or the next leaders, I would say. So Professor Niles, thanks so much for your kindness and contribution to the scientific and more importantly, educational community. And then by joining us and then spending your time with us. And thank you so much again. And the virtual podium is all yours now. And thank you again.
Thanks, Professor Moon, for that kind introduction and for the invitation to join in today's SynBYSS seminar. And it's a pleasure to share with you today, as I think, especially putting myself back in the shoes of trainee and early stage career exposures. The opportunity to hear the different journeys that more senior folks have traversed in their path to where they currently are, I think was super informative to me when I was younger. And I hope that, sharing a little bit about my journey might also be helpful to you as you think about your future. And I'll give you a little brief background of kind of how I got to where I am today. I'll preface it by saying it's not the most linear trajectory. but again, I think that there's value in nonlinearity in biological systems and in life. So I grew up in the Caribbean and had not really envisioned coming to a university in the US and the role that I'm in today. I knew I loved science, I knew I loved math and technology, but never really envisioned what future would allow me to pursue the things that I kind of felt passionate about even at that young age. I think like most of you, I was very fortunate to have really motivated and dedicated teachers who cared about our learning and cared about us applying ourselves to our fullest ability. And at times opening our eyes to opportunities that we didn't necessarily see on our horizons. And in many ways, it's really that combination that allowed me to see the possibility of even applying to a place like MIT for college initially, and then eventually making the decision to come here. So I did my undergraduate work here at MIT many years ago. We won't count them. And I started out in chemistry, and kind of really fascinated with molecular level understanding of how molecules work together to create and modify life. And those interests transitioned into kind of an interest in combining research in the life sciences and applied sciences with a career in medicine. So I decided that I would pursue a combined medical degree, PhD, for my graduate training. I didn't leave the area. I stayed around the MIT Harvard area for that for that combined training. The PhD component was back at MIT. And it's an experience I would not trade for another. I learned kind of how to think at a systems level and through a lot of the medical coursework. And at the same time, think in high resolution at molecular level through the PhD project that I was involved in. And so that kind of allowed me the range, the scale to see problems, how solving problems in molecular space could actually then eventually translate into kind of potentially human health impact. And I felt that that was maybe one of the key takeaways of the combined training. I had every expectation that I would go on to do a residency out of that program, but ultimately decided that the thing that I was most passionate about was the research and finding ways to excel in research that could eventually impact human health. So that led me to a postdoc out at Berkeley, again in a chemistry department, but thinking in problem space related to metabolism and also again a different aspect of molecular recognition and how one could think about designing systems to perturb biology at in small molecule space and eventually at protein level to then study function and then to be able to make those connections between altered function and larger scale impact on human health. And it's again through that experience of working in an area I had never worked in before. It turns out that's been a hallmark, I would say, of my career at each stage of the work I did as an undergrad, the work I did as a PhD student, that I did as a postdoc, didn't follow a linear progression. They followed things that I was interested in learning deeply about at the time, and recognizing that they could grow both my technical skills as well as my intellectual ability to think broadly across different areas. And so eventually it became time to take that next step. I kind of knew I wanted to be a faculty member somewhere and kind of working on something that would integrate kind of the totality of my experiences. And something that I was exposed to back as early as high school, tropical infectious diseases, stood out as an area where I felt I could have impact and where there seemed to be a really important need for the types of questions I was asking and the strategies I was thinking about in terms of how to tackle new problems in biological space. And so in a way, inadvertently, I got exposed to synthetic biology through that interest because we were working in systems. The malarial parasite that we work with is a non-model system where genetic perturbation was not a standard available opportunity at the time we started. And a lot of the work in synthetic biology was built on the ability to control at the genetic and other levels how you would design circuits that could achieve biological outcomes. And that framework kind of really heavily shaped kind of what the possibility landscape of solving what was an important fundamental question to how one studies these types of organisms really became then a problem that could be framed in terms of, how could you use synthetic biology to gain access to fundamental understanding to then further our ability to address a really major human health problem. And I happened to land at MIT where there's kind of a very open-minded community of people working on a broad range of problems. And I would say in a lot of ways, one of the homes of synthetic biology. So it was really, a pleasure to connect in with this community here to pursue the type of work that we've been doing. So all that's to say is I think maybe a key takeaway from kind of my experience from me to my younger self and maybe to you then as being much younger than I am now is follow the things that you're super passionate about. Learn to the fullest when you're immersed in your current situations. Keep your eyes on the horizon. The things that you do now may not always seem to be super relevant in terms of what might be gaining headlines and so forth. But they're informing the landscape that you can access to really define what your future trajectories and opportunities will be. And so everything you do, just put yourself into it 100%. And the world will be your oyster, proverbially. So I'll end there, and I'm looking forward to hearing from our rising star and the fascinating work that you've been doing.
Thank you so much. That's absolutely amazing talk. And then now I know about you better, and the journey is kind of remind me of one of my best students. He's also from another country, in Africa actually. His situation is so bad, including his home at the time was not in good shape, no electricity, of course, no internet. He lost 2 brothers because of all the African problem. I don't know that's because of malaria or not, but of course, there are too many problems in that continent. But he has one thing in common with you. That's basically the passion. And of course, the passion about potentially curing his mom. Mom still suffering from cancer. but anything related to medicine is his passion. And then he, endure all the difficulty, as a person of color, everything here, but he endured and then he finally joined Pfizer, the company. And then guess what? He developed the first COVID vaccine at Pfizer and then contribute to entire world saving a lot of people during pandemic. So I'm very proud of him. And I realized that was the reason I became a professor. So thank you so much. I mean, your journey sort of similar to what he achieved before.
Yeah, no, that's impressive.
Yeah. Thank you
Super
so
impressive.
much.
Yeah.
All right. So now the main speaker of today with a longer introduction. Dr. Jeremy Zhao is a research scientist from NYU Langone Health. His research focused on how to push the boundary of human disease research with a synthetic genome approach. He has led the construction of synthetic chromosomes in yeast, commonly termed SC 2.0, and integrated multiple of them into a single cell, a breakthrough published in Cell last year. At the same time, Jeremy leveraged his expertise in crafting large synthetic DNAs in mammalian systems to study human disease. He has developed the CRISPR, that's acronym, in other words, CRISPR engineering of episomes in yeast, again, the CRISPR system, which streamlines the engineering of large synthetic DNA in yeast with complex modifications and further integration. I'm sorry. So further integration, further integration into mammalian genomes. This novel approach has boosted his recent research studying the mechanism of a neurodegenerative disease called X-linked dystonia Parkinsonism by building a humanized mouse model. Beyond his research, he is also committed to education. In 2020, he established an undergraduate course at NYU called Build a Genome, where he engaged students in cutting-edge, hands-on research projects. He also mentors within the Build a Genome network to expand synthetic biology education across diverse institutions, especially under-resourced college and underrepresented groups. And that's, I believe, absolutely amazing effort, he already done, not just as the scientist, also as nurturing person for the younger generation. So Jeremy, thanks so much for your time today. And please take it away. It's all yours now. Thank you.
Thank you. Thank you for the super nice introduction. So let me start the sharing and make sure you can see the video and the slides. Is working well?
Yeah.
Perfect. Yeah, before I start, I just want to echo the word from Jacquin that I just checked my Apple Watch. When you give the short speech, my heart rate doubled. So because I just feel so excited about what you said. Just want to mention that my passion for research also started with biological engineering department at MIT when I attend the IGM conference in 2011, like more than 13 years ago. Probably like many of you who are watching this video online or watching this alive. During that time, there's nothing called genome editing or genome engineering. I just feel oh my God, we can engineer genome. It's so fantastic. And I try to learn how to do that. That's actually at the beginning of my passion for research. And also, as Jacquin said, nothing is linear in research because we are doing research, right? If we can predict the future, we are not doing science. We are doing some different things, or that's manufacturing, that's different. As a scientist, my interest or my passion is about how can we just push the boundary of genome engineering from the genome editing for genome writing. And I self-describe myself as a genome engineer because I'm particularly interested in exploring the genome complexity, genetic disorders about human disease, and about manufacturing with my unique approach of synthetic genomics. So today I'm going to share my research in the last few years and my passion and vision in the future. Since the discovery of DNA double helix, biologists, including myself, have been curious about what the genome is and how it functions. If we look at our keyboard, right, we have at least 26 keys for our input. But how many keys the genome need? Only 4, ATGC, right? How it is organized and how it's just encodes the whole life. It's just a truly thing in my heart I'm super interested in. And some 50 years later, much progress was made in the well-known Human Genome Project with the advanced technology in the genome sequencing or genome reading. In the past 10 years, CRISPR has made it easier than ever to manipulate the genome at a nucleotide level. It's truly lots of progress that happened. Looking forward to the future, I hope to bring the genome engineering to another level with DNA design and writing at a megabase level or even genome. To achieve that goal, we really need a comprehensive understanding of our genome. How are we going to do that? As Richard Feynman said, what I cannot create, I do not understand. Following that, my philosophy is understanding by building as a genome engineer. And specifically, I aim to dissect the genome complexity by designing and assemble an artificial genome in both yeast and potentially in the mammalian system in the future. In the computer science, engineering by writing has been pretty common, right? We are using lots of apps in our phone or in our laptop. People start with the design, right? We want to design a social media app, and then they will write a code following certain algorithm and finally build an app. But in biology, what if we can write a genome? In synthetic genomics, we also try to start with the desire, for example, produce medicines, antibiotics, or antibodies. For example, we can also do data storage in DNA produced by fields, agriculture, or organ transplantation. And we can also develop disease model for therapeutics. Then, can we just design a genome and engineer an organism in a single cell or mammalian system with a broad view of applications? This also will provide a novel perspective to understand genome in a bottom-up approach. Why is the time to think about that? One major reason is that the cost of DNA synthesis has dropped dramatically. This is a real quote from George Church Lab, who is also affiliated with MIT, I guess. It's more than 40 years ago. Only 10 nucleotide oligo cost how much? It cost $6,000 in 1980s, which is a big money during that time. And right now, it's just very cheap. So in 1980, it will cost more than 7 weeks to arrive. Right now, if you just order a primer, they will arrive the next day, right, on your bench. If it's delayed, it will cost 2 days, but it's very fast, and you are never worried about that. But during that time, it cost more than $6,000 more than 7 weeks, and it was not even guaranteed. And right now, we can just get much better skills to build a genome by doing the genome synthesis from scratch. But the biggest limitation of our artificial genome is actually not the cost, it's our imagination and our understanding of genomics. This is another figure showing that the DNA synthesis is from the Moore's law that dropped dramatically in the recent years. And we expect that the scale and the cost will continue to be reduced in the next 10 or 15 years. And following that, I'm going to tell you my research journey in synthetic genomics. So first, I'm going to talk about artificial genome in yeast, also called IC 2.0. And to build the genome, I developed the technology of CRISPR debugs And also the technology I'm going to mention briefly today called CRISPR for genome engineering. So what is CRISPR technology? I'm going to tell you more. And all my experience with yeast also motivated me to dissect the mammalian genome complexity in a mouse model. I'm going to talk about my effort recently in the genome engineering in mammalian system, actually in mouse for human disease. So first, I tried to engineer a genome in yeast of Saccharomyces cerevisiae, which is also called body yeast. It's a unicellular system and has been very well developed. For example, we are eating yeast frequently. For example, when we drink beers or eating bread, the yeast really make our life better. Just as like Ben Franklin said, the beers is a proof that God still loves us. Actually, we have a super long history of using yeast. This is the artwork from more than 4,000 years ago in 2600 BC in ancient Egypt. You can imagine that what they are doing right now here. So basically, they just store their plants or like food in a big tank or basket. After some time in the hot weather, what will happen? There will be fermentation, right? So actually, the people during that time found that there's some precipitation at the bottom of the can, and in the middle of the can, you will get some liquid. What's a liquid? Actually, that's beers during that time. So the people had already started to drink beers more than 5,000 years ago. And actually, this is the yeast that make our life better. If we check how the yeast are growing, they can grow as a haploid, which is there's only one copy for the yeast chromosome with the two mating types, mate alpha and mate A. So when they meet each other, they will fall in love and do the conjugation to form a diploid, which means that there's two copies for each chromosome if n equals 2. Under certain conditions, we can induce the sporulation and do the tetradissection to separate the spores generated from the diploid, where we are going to recover the copy of the chromosome or the genome from n equals 2 back to n equals 1. This is the life cycle of the yeast. Then the question remains how we can just design a yeast we can just do from scratch. So Saccharomyces cerevisiae is a genome containing 12 megabase. And how it's called, like, by the way, it's called sugar fungus of Saccharomyces, and cerevisiae just means that found in beers. So the question is how we are going to design the genome, how we are going to assemble the genome from scratch from short DNA oligo that was chemically synthesized. If we understand the genome, everything will be good. But if something we didn't understand, something new that we didn't expect, can we just do debugging? And also, can we just do some applications of the novel SC2.0 genome that we built from scratch? Let's lead to like 3 principles we try to do the genome design. First, to know how. My wife always told me that happy wife, happy life. And I also want to see the thing like happy yeast, happy life. I want to make the strain become healthy all the time. And also, since we are going to build a novel strain, right, we want to increase the genome's integrity by building a stable genome. And of course, we try to introduce some novel functions by increasing the genome flexibility to introduce several novel functions we can do we can use SC2.0 to study fundamental questions. This is how we are going to design the genome. And first, we start with the wild type genome. We try to improve the stability by removing all the retrotransposons and the mobile elements. We can see that whether a eukaryote can survive without any mobile elements. And what will happen if we reintroduce novel mobile elements into a genome that is retrotransposon free? And also, we remove all the tRNA gene to a special neochromosome. We also remove almost all the introns and recode the stop codon from TAG to TAA. Then we will have a free TAG codon that we can use to incorporate novel and unnatural amino acids into the protein that we can design in the future. Another feature I really like is that we insert log P site into the 3' UTR of all the non-essential genes, which will enable a powerful inducible evolution system called scramble. You can see that if we introduce recombinase into the genome will start shuffling because the recombination will be mediated by randomly any 2 log P same site, and this will generate thousands or even tons of pools of population that we can screen from genome evolution. This is how we are going to do in the future for the study. And also, this is the SC2.0. We are going to build them together. And finally, you may notice that the genome scale will be smaller because we remove several components from the wild-type genome. Finally, I want to say that we have a very fantastic international consortium that I was engaged, that we have an international team to assign the, to build the genome separately. Basically, each team is assigned with one synthetic chromosome to build from scratch. It has been done like recently, actually, we recently published a series paper that we finished assembly of all the synthetic chromosomes individually in their own strengths. But one challenge still remains how to consolidate multiple synthetic chromosomes into one cell. For example, the team of synthetic chromosome 1 have the same one in their strand, but all the remaining chromosomes are still wild type, which means that more than 90% of the genome is still wild type. As a research scientist right now, I'm leading the effort to try to consolidate all the XC2.0 chromosomes together to be one strain. You can imagine that there will be the strain with 100% artificial genome built by human beings. How we are going to do that? So one thing is that, for example, this is a strain with synthetic chromosome 2, and this is a strain with synthetic chromosome 6. One thing we can do is that we can just put them into different yeast mating type, mate alpha and mate A and mate alpha. And in their counterpart of the synthetic chromosome, we can integrate the pGAL promoter in front of the centromere. pGAL promoter is one of the strongest promoter in yeast that can be induced by the media containing galactose, right? So we can just mate them together. This is haploid, mate A and mate alpha. After mating, we will get the diploid cells, which is heterozygous because the strain containing both synthetic version of the chromosome and also the native version, right? In this heterozygous diploid strain, we can induce expression of the pGAL promoter right in front of the centromere. You can imagine that just like a bullet train, the transcription machine will travel through the centromere very fast. And this will block the binding, occasionally block the binding of the canonical to the centromere. This will cause chromosome destabilization. And the yeast will say, oh my God, what happened to my genome? I lost my chromosome. And the yeast will try to do the recovery by make another copy of the chromosome. And you can imagine that there's only synthetic version available in the cell. And the cell will make another copy, which means that strain will become homozygous. In the other words, all the genome, all the chromosome will be synthetic. And then we can just induce the superylation and do the texture dissection to get a final strain containing more than one synthetic chromosome, for example, SYN2 and SYN6 here. You can imagine that we just go through from the haploid, heterozygous diploid, homozygous diploid, and finally back to the haploid. And by iteratively process, we can just consolidate different synthetic chromosomes together. And finally, we get the strain with more than 6.5 chromosomes together. This is account for almost half of the genome has become synthetic. After we get this strain, we just super curious about how the genome looks like, right? So I just use the technology called a pulse field gel. Basically, we can just run a gel similar to agarose gel and stain the genome by itself. So we never break the DNA, we just see the genome. In this process, we can visualize the full length of each chromosome. And you can see that we can just check, oh my God, the strain containing successfully consolidated synthetic chromosome, which is like a smaller compared to the native counterpart because they are migrate a little bit faster compared to the wild-type strains. And also we use the whole genome sequencing and Illumina sequencing together with nanopore, confirm that strain carries the correct genotype. There's no mutation in happening in this process, which means the endoreduplication intercourse process is really working well. I just want to highlight that in the final strain with 6.5 synthetic chromosomes, there are already more than 6,000 designer modifications that we have introduced. For example, we already introduced more than 1,000 log-P same sites. This will generate a huge population and a big diversity. And also, we already recoded more than 400 stop codons of TAG to TA. And this is, this is the powerful of the synthetic genomics. We can design the genome from scratch. We just cannot do that with CRISPR, right? CRISPR has been powerful, but how can you imagine that you want to do 6,000 rounds of CRISPR to reduce modification into artificial genome? Well, I believe students will never graduate, right? They do not do the experiments. After we get the strain, we're also curious about how the synthetic genome is organized in 3D trajectory. You can see that this is the picture we get using the Hi-C. They just connected together by bound by canonical at the one end, and also their telomere was extended naturally to the periphery of the nucleus. they have the similar trajectory in 3D model and the wild-type genome. Again, we try to increase the stability of the genome to remove the resource transformer element. Meanwhile, we keep the strain be healthy, behave just like wild-type, and keep that in mind. We already have more than 6,000 modifications we can introduce through the synthetic genomics. So with that, I just to summarize my first part is that I built the artificial genome of SC 2.0, and we are going to do so many evolutions using the scramble system. This will be the super yeast for biosynthesis. Of course, we are faced with some bugs that cause the strain fidelity, and we use a technology called CRISPR debug for genome scanning. I didn't go to the detail for the method, but we have the method to build the genome, assemble the genome, and also repair the genome. In the second part of my talk, I want to just talk about my current research and future research about the human genome. So the yeast genome is only like 12 megabase. How large is the human genome? It's 3 billion long. So what's the meaning of 3 billion base pair long human genome? You can imagine that if I have the magic, just like Harry Potter, to drag my DNA out of the cell of the brain and put it into the hands, I can tell you the length of the human genome is more than like 2 meters long, which means that if I hold that in my hand, it will drop to the ground. It's just simply a miracle that our human cells can pack our 2 meters DNA into a small nucleus that we cannot even visualize. And more than that, we can just put it into a small space and regulate the expression of each gene normally. This is just such a miracle that I just really want to study how the genome is organized and regulated using my approach of synthetic genome. And another thing I really want to highlight is that around all our genome are organized in a small space, but only 2% of the genome are encoded for protein. What else? All of them are basically like regulatory elements. What's the rest? So how we are going to do that? And another thing I want to highlight is that for the rest of the genome, we usually call them as junk DNA because they are not encoded for protein. But actually, they do have some signs of biochemical functions. We also know that non-coding genome is extremely important because the vast majority of the trait and disease-associated genetic variants fall into these non-coding regions as identified by the GWAS. With a CRISPR, we can just pinpoint each nucleotide variant, but we really cannot do, for example, 1,000 of modification. What's their rule? And we just cannot building a genome from scratch. So this is why I want to do my approach, do my research to decode the regulatory function by synthetic genomics. This is one of my favorite picture, and we just describe the non-coding genome as the genome dark matters. We know it's there, and we know it's play critical roles in the function of our genome, but we do not, or we cannot fully understand what it is doing. How to decipher that with genome writing? In Book Lab, we design and build big DNA in our favorite system, yeast. I guess you have been convinced that I do have lots of technology to build the big DNA in yeast cells, and we can produce the assembly-tron, which is also called payload. Then we can try to jump out of the yeast. We can just deliver that into the DNA, into the mammalian cells, such like the mouse embryonic stem cells. Then we can just examine the DNA we deliver that directly in the mouse embryonic stem cell, or we can generate mouse as an in vivo model. We can also differentiate under certain pathways where we can just direct our design and assemble further based on the characterization we learn from the modification that we learn from our first build. We can use the yeast to build this synthetic loci. This allows us to build a assemblage completely de novo from artificial synthetic DNA fragments. We can just stitch them together, like for example, 3 to 4 kb fragments of synthetic DNA into an entry vector that allow the maintenance, assembly maintenance, and propagation in yeast and bacteria. Alternatively, we can also repurpose the existing sequence, for example, in the back library. We have so many human and mouse genome fragments there. We can just assemble them into the same sector vector and do the genome editing using CRISPR system. So basically, the assemblage will exist as the episode in yeast. We can further engineer to incorporate thousands of modifications or variants as the CRISPR system. Keep that in mind. CRISPR or CRISPR engineering of episome in yeast is doing the genome engineering in yeast, not mammalian cells. This process is completely offline, which means that before we deliver into the mouse embryonic stem cells or human cells, the sequence has been verified. And you do not need to worry about the offline effect, non-specific technique at all, and you are 100% sure what you are delivering is the sequence that you want. Not just a wild type sequence, but any sequence that you can imagine you can design, including, for example, deletion, insertion, or mutations in the gene regulatory elements. Finally, we can just extract them from yeast, amplify in bacteria, and prepare for the transfection into the mammalian cell. We usually start with the marker cassette. Basically, we just integrate the marker cassette to delete the original native sequence. This can be very long, can be like up to a few hundred KB. And then we can just overwrite the marker cassette with a DNA payload. Again, this is not native. This is contain the modification as we designed in the yeast and engineered by creepy system in yeast to generate a series of different mammalian cells carrying any kind of genotype you can imagine. And also you can just do the experiment in parallel, which means that it can generate lots of variants from the isogenic background. Basically, they're actually siblings, right? They are from the same parents. This really facilitates our characterization of the variants that we want to study. And using this method, we can just build a semblance, further edit them, and deliver to the mouse embryonic stem cell and generate mouse. We can just even delete the mouse region and replace that with a human DNA. That will be our humanized mouse, right? Using this process, we already engineer mouse model for disease. For example, today I'm going to talk about humanized mouse brain. Basically, we just make the mouse brain carrying human version. It's a basically mouse model for XDP. And also we, at the same time, my student, Miwash, is leading the project to build the humanized immune system, a mouse with human immune system. And also another student, Sunny, is trying to build the mouse model for iron metabolism dissection, basically the disease associated with blood. So today I'm going to talk about what is XDP. So XDP is an X-linked inheritable rare disease. It's a severe Parkinson's disease. It stands for X-linked dystonia Parkinsonism. This inheritable disease is uniformly fatal and has no cure or even no treatment right now. It's really terrible for the patients. It's rare and really very terrible. The patients carry an extra SVA resource photon insertion deeply in the middle of the large intron of the essential gene called TEF1 in X chromosome. So basically, people may think a resource photon insertion into the intron has no effect because it will be spliced out, right? But turns out to be this is not true. So if we just check the diagram of a family, you may find that there's kind of one super rare event happening to the grandfather. Fortunately for their daughter, they are fine, but very unfortunately for their grandson, half of them carry the disease. This is a typical thing happening for the X-linked chromosome, right? How we are going to build a mouse model and figure out the mechanism why this resource photon insertion cause the severe disease of XDP. And you can imagine that we have the tool to build them, right? So basically, we start with the human version, human TEF1 back that generated directly from the XDP patients with a length of almost 100 KB. We just repurpose the host fragment into the mouse and insert the mouse arm for the for the future insertion of the mouse genome. Basically, this fragment will carry the downstream of the half of the human genome or human version of the TEF1. And you can imagine that we can mousify that. Why we need to mousify that? Because we are only curious about SVA insertion, right? There are some coding sequence different between human and mouse. We do not affect the protein. We just want to study the regulatory element of this gene. In the other words, we mousify the humanized TEF1 by changing all the amino acid from human to mouse. In the other words, the TEF1 genes or the hybrid version will carry mouse protein sequence, but human intron and exon structure. Of course, with creepy system, we can build lots of variants ease. For example, the one simply control experiment we can do is just delete the SVA, right? Just to see whether our hybrid human version can function or complement the deletion of the mouse version in the mouse cell. Again, everything is done in yeast. So you do not need to worry about the non-specific effect at all. I guarantee that everything will be correct. And then we can just do the assembly, build the variants, and finally deliver into the mouse. And into the mouse, it will carry the 5' end mouse type 1. In the other word, the promoter region or the enhancer region are 100% native. The expression of gene is not affected except the resource sponsor insertion of SOV. We get a mouse immune stem cells, and luckily we get a mouse. How it looks like? You must be very curious, just like I am. So this is the wild type control mouse, right? This is the XDP male mouse that's showing the carrying the human version. See how it looks like. So one thing you may notice that first they are smaller, right? So they are smaller compared to the wild type control. And another thing you may notice, you check the tails, you see that they are not moving very naturally, right? For the wild type one, it's just rotating very freely. But for the type 1 mouse, it's just like reach. They just cannot move, cannot control the movement of the tail and smaller. It's just like hanging around, wondering who I am, what I'm doing right now. This is a typical pathology that we observed for the patient with Parkinson's disease. And what will happen if we just integrate the humanized version, but without XTP, without the SOV insertion? You can see that the deletion of the SOV insertion completely rescues the defect, which means that the defect is caused by the version of the SOV insertion instead of our engineering approach. Of course, we have so many quantitative data. For example, compared to the wild type male, the XDP males showing basically they are losing the weight, showing like smaller weight compared to the wild type. And also, very interestingly, we also get the heterozygous female, right? So how they're going, you can see that the heterozygous female behave. At the same time, we got to get the male with SOV deletion. You can see that with the deletion of the SOV, it's completely rescued the defect, just like the wild type. One thing I didn't show, I just mentioned that I didn't show that today, but I can tell that we also get the heterozygous female with the XCP version type 1, and also the native version S1 type 1. It behaves very well, just like a wild type. Finally, at the end of my presentation, I just want to show you my data of my favorite experiment is that kind of just like called cat working for the mouse. It's really funny that we try to monitor the cat working for the mouse to see whether it can just work like a cat. This is how it works. So basically, we put the mouse at one end and try to attract that to move to the other end. And we have a camera at the bottom shooting up above to just record the gait of the mouse, basically left front, right front, rear left, rear right, to just record that. And we can just visualize that and also quantitative that and quantify that. So this is our data. This is a male mouse. Wild type control is working so well, smoothly, and this is our XDP humanized mouse. You can see that it's working slowly and the gait was completely messed up. I promise I didn't manipulate this video. I play the video at the same speed. They are just working slowly with abnormal weight and abnormal gait. So finally, I want to summarize my talk today and just to share, I'm super excited to share my interest in genomics. So as a genetics, I'm super interested in doing the artificial genome to figure out what's the fundamental question we can answer in synthetic genome, in genomics field. And of course, to answer these questions, what kind of technology we need to develop to answer these questions. And following our research, how our discovery can benefit human health. And following this goal, I talk about artificial genome in SC 2.0, and we're going to do lots of evolution and scramble. Of course, there will be the super yeast for biosynthesis. And of course, in the future, I'm moving forward to the mammalian genome synthesis, try to dissect the complexity associated with human disease. And for example, we already started to study lots of neurodegenerative disorders, including Parkinson's disease and Alzheimer's disease right now. And my biggest interest right now is try to use a building approach to study or map the variants to functions called V2F. And finally, I already developed lots of technologies, for example, CRISPR debugs for genome scanning to repair the genome in yeast. And also I developed a creepy method for big gene assembly and engineering in yeast, which can be delivered into the mouse immune stem cells and potentially human cells in the future. Actually, not just the mouse and human, any kind of mammalian cells, we can do genome engineering and genome writing in the future. Of course, I want to move this direction to be high throughput and automation. So this is the thing we are doing right now at the Boca Lab, try to make this process in parallel, high throughput, and do the design in silico in a high throughput way. So finally, I want to give my special thanks to Jeff And also the project of the XDP is co-mentored by Shen. And also I want to give special thanks to the SC2.0 team, Luciana, Wimi, Laura, and Max. And also we have a fantastic S2.0 consortium with many collaborators from all over the world. So XDP project is a collaboration with Wimi and Priya. So we're leading this project together. I want to give my special thanks to Ryan, which mentored me how to do, mouse induced stem cell culture. It turned out to be that the people who are doing TC really hit me at the beginning because I'm doing yeast. So they are super worried about the contamination of yeast in their TC room. But so far, they are so good. Finally, I want to give my special thanks to my students. I teach the build genome class at NYU since 2020. Actually, they are amazing. My students did more work than me right now. I just use that data. And also, a few months ago, I was awarded as a team the Elizabeth Jones Award for Excellent in Education by Genetic Society of America, together with Jeff. Special thanks to Sandy, Camila, Cindy, who contribute to the IC 2.0. Again, like my passion of synthetic genomics started with IGM. Actually, I'm still doing that contribution to the IGM by working as a judge. I'm really happy to see many of you will continue to join our community and to inspire more and more students. So finally, I also want to give thanks to my foundings. Special thanks to Moon. My research is really effective, really inspired by Richard Feynman. So Richard Feynman is a great communicator. His teaching and lectures on physics inspired tons of students around the world, right? So he's more than a scientist, he's a communicator, just like the Dr. Moon is doing right now. He's a fantastic YouTuber and trying to broadcast science to a big audience to inspire more students and more scientists to doing synthetic biology. This is something I cannot agree with more. And with that, I'm happy to answer any question and stay here for a while to any kind of discussions.
So thank you so much for your kind remark at the end.
Thank you, Moon.
And also, amazing talk. All the amazing technology you developed, one thing struck me. You said the happy life, happy wife, and happy life, and happy east, and happy east is. But I need to say now, happy young people, happy community in the future. So that's one thing I want to create new. So absolutely brilliant talk, amazing tools. I love your video with the mouse. That's amazing video as well. So let me ask the Professor Niles whether he has any comment or question first.
No, fantastic work, Jeremy. I mean, this is like really beautiful.
Thank you. Yeah. Thank you, Jacquin.
Yeah, yeah. And I have a question on your like in your yeast system in terms of the LOX-P recombinations and how you're planning on leveraging those going forward. Can you comment on like kind of the kind of chromosomal translocations and how you're controlling that so that you get, you don't create variants that might be defective? Is there a strategy that you envision that'll minimize that?
Yeah, of course. So basically, like Jacquin is talking about the scramble system. So it's one of my favorite feature in the IC 2.0. So this is a slide I used it before. Basically, like when you integrate in the final IC 2.0 genome, it will contain more than 3,000 of piece inside. This is a big tool. So actually, I already get some preliminary data. I can show you here. Okay, here we go. So basically, like in the IC in string 6.5, in the string with half genome synthetic, it's only like more than 1,000 log piece inside. We already started to try to turn on the scramble system. So the scramble system is not on all the time. It's inducible. So basically, we just transform the query combines and also have a switch called estradiol. So we use the estradiol as a drug to turn on the scramble. So it's not always on. And also, like after that, we just generate a super big diverse population, and we try to map them. Actually, in just one round of scramble, we just detected more than 260,000 events. It's really a big pool. It covers all than 1,000 log piece inside. So I want to say that this is really a big population. If you do not have any selection, you will get lots of data, right? The point is that what kind of direction you want to do the evolution. So basically, like one direction is that we can just do the evolution for certain, for example, producing more biofuels. This is the thing we're doing right now. So we can just implement the biofuel synthesis pathway into the yeast, and we can just integrate another called a reporter for the synthesis of biofuels. For example, GFP to JGFP, something about that. And then we can just induce a scramble, random thing, and we just use the cell sorting to dissect the yeast strains, yeast involved strains with a higher production of the biofuel, and we just check out and do a sequencing to see whether we can learn this in this process, why bring the production of the biofuel. Another super interesting biological question that I'm doing that I will try to do in my new lab is that what's a minimal genome of yeast, or what's a minimal genome of eukaryote? So we know prokaryote and eukaryote, right? So how we define the prokaryote eukaryote? Based on the observation, right? We see that eukaryote has the nucleus, prokaryote not, cell structure, blah blah blah. But I'm genetics. I just want to know like how the nature or how the God define eukaryote or prokaryote from the perspective of the genome, and how to tackle that question. My perspective or my approach is that can we just minimize the eukaryote genome to see what's the essential point, essential set or combination of the genome that still can keep yeast yeast, probably not yeast anymore, but a yeast, something about that. So the thing we can do is scramble, right? So the advantage of scramble is that once the genome fragment is lost, it will not come back. So basically, like from the long term, actually the genomes intended to shrink. Right? We just kind of do like a lot of batch transfer and try to isolate single colony, like smaller genome. Another round of scramble, we will get a different kind of a tree of evolution to see how the genome scramble will happen. So this is what we're going to do with the genome scramble in yeast for chromosome rearrangement. Yeah. I hope that answers your question.
Yeah, no, that's very helpful. And and then like kind of can you comment on the resolution of the size fragments that you envision you can move around this way?
You mean the resolution? Actually, the resolution can be very high, because for whole genome sequencing, we get a very deep high throughput, and we can just take out the reads that still containing the log P same size fragment, which is only 34 base pair, and then we will get different reads outside of the 34 base pair. We can just align to the genome whether we can get a normal location or different location. Right, so if it's wild type, it will align to the original location. But we may find that okay, the one side of log P same log P side is from one genome, and another side of the log P same size sequence actually from another genome. Then we will know how the genome rearrangement happens. Actually, we already have the algorithm to dissect this very well.
Right.
Yeah. For the resolution
and looking.
Yeah. And also like there's one limitation, to be honest, is that the scramble can only happen between log P same side. So basically, when you integrate log P same side into the gene, so basically we are not introduce random mutation into the coding sequence of the protein. We just do like a gene level. We didn't change the gene itself. For example, we can call it transcription unit. For example, TU elements transcription unit. We just do the TU rearrangement, deletion, inversion, or deletion. We didn't change the protein itself. Yeah.
So, Dr. Niles, do you have any more question or comment?
Yeah, if there are other questions, we should allow those. And I can also, I have a few more questions for Jeremy that I can also ask him offline.
Yeah, please do so. Yeah, please do so. Yeah.
Yeah.
Let me check that. Okay, here we go.
Yeah, I do not see any more question from the audience for now, so you can ask more.
Right. And then in terms of the size, like fragments that you can move into, like especially in the mammalian engineering mammalian cell engineering you've been doing. So it sounds like you build in yeast and then you move into the mammalian cells. Like what's your cargo size limitations that you're running up against there?
Yeah, so basically like the whole procedure is that we build, engineer, and deliver, right? So first, so we are talking about skills. So currently we already successfully integrate like 100 KB, for example, using there are two detailed methods I didn't talk about. One is called BigIn, another called mSwap-in. So BigIn is a Cre-Lox base, and mSwap-in basically CRISPR-Cas9 based. So we have successfully routinely integrated like more than 200 KB right now. Basically, 200 KB is a kind of limitation. So what brings the limitation? Why we have this kind of limitation? It's the next question, right? Turn out to be like building yeast is really straightforward. I can build the whole genome. So basically, that's not the major thing. And CRISPR engineering, we can do like a 10 editing at the same time right now in the genome, and we can do like 5 editing for the modeling sequence at episode. We can do the multiple round editing. So the editing is also not a critical step. It's not a limitation step.
Right.
Sorry. So the current limitation is the delivery, which is that how we are going to use nucleofaction or transfection method to deliver the payload, which is the DNA, into the modeling itself. Our current limitation is 200. I tried like more than 200, for example, 250 or 350. It turned out to be that we will get some colony, but it's just a fragmented DNA. So it's just broken. That's why like one thing we're thinking about right now is that we need to develop a new method, which is not cell-free. So current method is cell-free, right? We need to prep the DNA using like midi-prep, something from the E. coli itself. And we need to handle the DNA by pipettes. Although we are using the white ball pipette tips, it's helpful, but not something I think we need to continue to move forward. So currently we are thinking about, can we just deliver, like for example, 1 mega DNA without taking out of the E. coli or yeast? Just can we just do some sporoplast structure or even like a lipid structure, like with the DNA inside, and fuse that structure together directly with the mouse improved stem cell? That will break the limit. So I believe that's the direction we can do. So one preliminary experiment I already start to think about is that we can even isolate the nucleus of the yeast, right? Not the whole cell, but just take out the nucleus of the yeast. And you can imagine the nucleus is actually a lipid structure that carries DNA, right? We can then digest the whole genome out, just degrade the whole yeast genome, but keeping the payload, and just deliver the nucleus directly into the mouse stem cell. That will break the limit in the future. So that's kind of the thing we are going to do. But currently, with like in practice, we can do 200 kilobase pair at the same time easily. Last thing I want to mention is that mSwap-in process is having iterative. Basically, we can just switch the marker, basically integrate 200 at first time, and then another 200, step by step. It's kind of time consuming, it's laborious and time consuming, but we can do that.
Yeah.
Any more question, Professor Niles?
No, this was great. I mean, and really exciting work, and
thank you.
Keep it up. Keep pushing the limits.
Thank you.
Yeah, that's the thing. Making me excited. I think
so,
you know.
Go ahead.
Yeah, I agree. I totally agree with Jacquin, because like when you talk about 200 KB, it's a level of human genes, one gene. Right, human genes huge. Like 100 KB, just one gene, one regulatory elements. We are still really far away from like 2 genes, 3 genes, or even the chromosome. We are really far away from that. So how going to do that is my goal. Yeah.
So actually the Professor and I actually asked some question I want to ask, and then I also see the central time 11:08, and then my time already past 1 a.m. So I will ask briefly one question. So this is amazing. Whenever I also need to be more excited, because now I am at the JCVI. That's a very famous genome synthesis and then creation of whatever new organism type of institute. To me, I mean the JCVI is the first paper about microplasma, the the creation of whatever in a cell. To me, that's not creation because they still copy original native, template and then just copy but gigantic scale and then made the first synthetic genome and then reboot. And then yeast is probably the much more complex. Actually, E is much more complex because they have, the yeast has more, copy of the genome or the chromosome. And then you already mentioned, some of the synthetic multiple chromosome thing already happened. But when do you think entire synthetic yeast, chromosome, first synthetic genome, complete yeast system will be happening? And then what is the current more challenges we need to overcome? Very simple word. I mean, I just want to know, potentially encourage more people to do those kind of amazing collaborative work. But I want to know, I mean, if that is the 100 years, that's kind of depressing. But if that is the 10 years, that might be very encouraging, right? So I want to know just how more years, I mean, we will see those things.
At the rising star, somehow I'm going to start my new lab soon, right? Hopefully, looking for a job and start my new lab. So I see 2.0 strain with 100% synthetic will definitely be my first project in my new lab. So I'm going to take it to my new lab. So it will not be now. It will be soon, not very long.
So that means, I mean, not just 50 years, probably 10 years time frame.
Well, I do not want to stay here for another 10 years.
Good. All right. That's good. So let us close because that is the 11 Central Time, and then my 1 a.m. Already passed. So amazing again. So that's absolutely amazing. Actually, you make me stay up. I mean, otherwise I already fall asleep because I didn't sleep much last 3 days. I mean, actually. So thank you so much all for joining and staying today. And we'll meet again on June 13th, Thursday, the same time, the same Zoom link. Actually, I will also still in Korea, so that means I need to do it again, the midnight to pass the 1 a.m. thing. We'll have the Professor Michael Alloyd at Caltech, also at the HHMI, and Brendan Camelato, also at NYU. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in joining us. And thanks, and I will stop recording. Just give me 1 second.